Hi, I'm Bob Naviaux, and I'm going to be giving a talk today for the Bench to Bedside presentation on the general topic of mitochondrial targets and defenses in the time of COVID-19. So my talk is going to be on the multi-system fatigue and hypometabolism that is coordinated with virus resistance that ultimately regulated by mitochondrial function. So we call this the two-edged sword of cell danger response and mitochondrial function. So the talk is divided into three sections. There's a brief introduction about hypometabolism and the leak of ATP out of cells. There's a part of this that relates to a paper that we recently published with Bhupesh Prusti's group on human herpes virus 6 and mitochondrial fragmentation. And then a brief look at SARS-CoV-2 and mitochondria. So turns out that mitochondrial hypometabolism is a universal biological response to life-threatening stress. And so cousins of mitochondria, bacteria, undergo what's called a persister cell transformation. And that becomes that allows them to reduce their metabolic exchange with the environment and become less sensitive, and because they're taking up less potential toxins like antibiotics. So that happens in Lyme disease and tuberculosis. But also embryonic diapause, where fertilization occurs and then the embryo arrests after implantation for weeks to months, and in the case of plant seeds, that arrest of the embryo can take place for years before the development is restarted after germination. So hibernation is another kind of hypometabolism. Torpor is a kind of decrease in basal metabolic rate seen in hummingbirds. Estivation, I won't go through all of these, but Dower is an important one that is a kind of persistence metabolism studied in nematodes, but related to the metabolic phenotypes associated with caloric restriction that lead to longevity. So here's a cartoon of cells that actually have channels in the membrane that can be opened or closed that will release ATP. So ATP inside the cell is a good thing, but ATP outside the cell is one of the most fundamental and ancient danger signals that actually recruits neutrophils and causes inflammation. So we asked the question if ATP leaking outside the cell could be a potential regulator of metabolism. What would happen if we injected extracellular ATP into a mouse? And it turns out when you do that with, and if you just inject saline, you get no change in body temperature. But if you inject ATP or ADP, there's a 6 to 8 degree drop in body temperature in just 30 minutes. And then that turns out to be associated with a 74% drop in basal metabolic rate measured by oxygen consumption rates. By indirect calorimetry in the whole animal. This is because mitochondria are the major heat generating organelles in the body. And so when they decrease their overall metabolism, heat generation in the animal is dramatically decreased, as well as many other things. So how does that relate to some of this antiviral work? So we did some work about human herpes virus 6. And here's a little fluorescent in situ hybridization karyotype that shows HHV6 can actually integrate into the telomeres of cells. It has a 4-stage life cycle of primary infection, latency, incomplete reactivation, and complete reactivation. So the interesting thing is that nearly 100% of us have received HHV6 in normal mother's saliva. And it will infect about 1 in 100,000 cells, but 90 to 100% of us by age 3. So 99% of HHV6 infections are silent, but about 1% can cause a childhood fever called roseola and 6th disease, and can lead to febrile seizures on occasion. And I've already mentioned that it integrates into the telomeric region of genomic DNA and can remain latent for life. Okay, but under conditions of stress, will reactivate partially. And that's what I'll be talking about. So occasionally, 1% of people in the world actually have a germline transmissible, copy of HHV6. And I mentioned already environmental stresses that will activate it. And immunodeficiencies can actually result in dramatic upregulation and reactivation and life-threatening disease. So now about mitochondria and HHV6. So here's a photomicrograph with mitochondria stained green, and the nucleus is in blue. And you can see this is a nice filamentous network in cells that do not have an integrated copy of HHV6. And there's also a nice filamentous network of cells, of mitochondria in cells that do have an integrated copy. But under conditions of stress, in this case, we've stressed the cells by treating them with a histone deacetylase inhibitor called trichostatin A. And we can so it turns out that that doesn't have much of an effect if the cells have no HHV6, but if they have HHV6, there's a dramatic fragmentation of the mitochondrial network. And what's that associated with? Well, we can measure ATP in the cells, and if the cells have no integrated HHV6, and we stress with TSA or try to lower them in galactose instead of glucose, then there's a decrease from control, but it's much greater decrease when the cells have HHV6. And now what if we now take the secreted products of the cells who have had their mitochondrial network fragmented, and then use that supernatant of cells to see if we can transfer the ability to resist viral infection. And it turns out just using that cell culture supernatant and treating other cells, we can actually dramatically reduce the ability of influenza RNA to influenza replication in those cells, those treated cells. And we can also have adoptive transfer of that antiviral resistance in using a challenge with a DNA virus, herpes simplex 1. So let's connect this to chronic fatigue syndrome. So if we look at the serum of patients with chronic fatigue syndrome, first, we have control cells that have a nice filamentous mitochondrial network. But if we treat those cells with the serum of patients with chronic fatigue syndrome, the network fragments, whereas a healthy control serum does not fragment the network. And then something else very interesting happens. With that fragmented mitochondrial network comes the ability of those cells with the network that fragmented to dramatically inhibit both RNA and DNA virus replication. So here we have, so the one position means 100% of control viral replication. In one case, there was actually an increase, but in the vast majority of chronic fatigue syndrome serum there's a decrease. And that also happens for human herpes simplex virus 1, whereas treatment with healthy control serum did not decrease this in the vast majority of experiments. So to summarize those results, here we have a plate of cells that have integrated HHV6 that's been reactivated by stress. We can take that supernatant and treat cells that do not have HHV6, and then test them for their ability to either be permissive or non-permissive for viral infection. So in the A to B sample, these mitochondria are fragmented, and they have decreased aerobic energy production, but it comes with the ability to now resist viral infection. And in the case of without HHV6, when we expose those cells to stress or use control serum, those cells now do support infection. So how do we put this all together? So in the stress cells where the cell danger response has been triggered, the cells that have HHV6, an integrated copy that has been previously latent, those cells can use that as a kind of barometer of environmental stress. And so lower levels of stress are able to fragment the mitochondrial network. That leads to ATP leak and extracellular ATP increases. Well, the consequence is, of course, intracellular ATP decreases. So the cells become less able to generally metabolize, grow, and perform their normal housekeeping functions. In addition, there's an increase in what we call oxidative shielding, which is the production of superoxide and hydrogen peroxide in these responder cells. But when the CDR is off, none of those things happen. And the cells remain sensitive to viral infection. Let's move then finally to this interesting story that's emerging about the novel coronavirus, SARS-CoV-2, hijacking mitochondrial proteins. This is a nice paper that was published in June by Keshav Singh's group. So SARS-CoV-2 is an RNA virus that makes 29 different viral proteins. But interestingly, those viral proteins interact with 20 mitochondrial proteins. Those include proteins, so non-OXPHOS proteins, but still mitochondrial proteins like prohibitin, but then a number of complex 1, subunits, a small ribosomal protein or ribosomal subunit proteins, as well as cytochrome C oxidase 2 subunits, some fatty acid oxidation proteins, CoQ synthesis proteins, and even the mitochondrial antiviral sensor itself. So what are some of the challenges that we see moving forward for, in general for all patients, but this is also of particular importance for patients with mitochondrial disease because they're starting out with an overall function that is already reduced from normal, and yet coronavirus will specifically attack mitochondria in interesting and complex ways that I won't go into just now. But 35 to 53% of the post-COVID-19 patients have a persistent life-altering fatigue that lasts, more than 2 months. That's also associated with mitochondrial dysfunction. And I'll mention that plans are in the works for a small trial of a form of oxaloacetate, a Krebs cycle intermediate that's the enol form, also chemically identical to hydroxyfumarate, that is actually available commercially already as a supplement. It's called Benagine. So that's going to be a study that will be done to see if we can't help some patients with this, life-altering fatigue recover their pre-COVID functional capacity. And now many of the patients that get stuck in this post-COVID fatigue will develop myalgic encephalomyelitis chronic fatigue syndrome, and that requires the diagnostic triad of fatigue for at least 6 months, but it can go on for decades if it doesn't recover spontaneously within the first 2 years. So it results in fragmented non-restorative sleep and post-exertional malaise, in addition to episodes of brain fog and postural orthostatic tachycardia syndrome and dysautonomia. And then finally, perhaps something that people haven't thought much about yet, but because of the way that coronavirus works and targets mitochondria, we see that there's potentially a risk for increased numbers of children who will develop autism spectrum disorder after these pregnancies. So this is yet to be determined. We won't actually even know this for a year or two, but this is a concern. So I just want to say thank you to our financial supporters for this research. So the UCSD Cristini Fund, UCSD Mitochondrial Research Fund, Mito Walk and Roll, and over 2,000 grassroots anonymous and grassroots support organizations and people, and anonymous donations, in addition to several found other funds, JMS, Lennox, Khosla, and Cohen. So I'll end there, and thank you very much.